everyone. Today we are going to discuss topic biosynthesis of nanoparticles. First of all, I would like to extend my sincere thanks to Physics Department, Namchi Government College, who gave me the golden opportunity to do this wonderful project on the topic biosynthesis of nanoparticles, which helped me to do a lot of research and I came to know about so many new things. I am really thankful. So let's start with our topic. Topic of the day. Introduction, definition, approaches used, nanoscale structure synthesis methods, biological system for nanoparticles, characterization, application. Nano and technology. Coming to introduction, nano and technology means the understanding and control of matter at dimensions of roughly 1 to 100 nanometers. Nano is the idea first introduced by the scientist Richard Feynman in the year 1954, and the name of the coined of nanotechnology by the author Norio Taniguchi in the year 1974. These all are some examples of nanometer in 14 nanometer, 10 nanometer, atom of 0.1 nanometer, DNA is 2.5 nanometer. What is nanotechnology? Nanotechnology is the art and science of manipulating matter at the nanoscale. It allows the placement of small structures placed with precision, simplicity, and its low cost. Nanotechnology is the study of small microscopic things. It is an advanced in future. Approaches for nanoscale structure. There are totally two approaches will be there, such as top-down approach and bottom-up approach. Top-down approach means breaking down of the bulk material into nano-sized structure or particles. In bottom-up approach means build up material from bottom, atom by atom or molecule by molecule. They are ranges small component into more complex or layer by layer. Various nanosynthesis methods. Nanoparticles are being synthesized by various methods like physical, chemical, and biological methods. The physical and chemical methods are costly and toxic to the environment. Physical method involves the uses of costly equipment, high temperature and pressure, large space area for the setting up. The physical method, there are several types will be there, such as ball milling, thermal evaporation, lithography, vapor phase. The chemical methods involves the uses of toxic chemical which can hazards to the person and the environment. The chemical methods are precipitation method, chemical reduction of metal salt, electrochemical method, Sol gel process method. In chemical method, mostly used of vapor pressure method. Third one, biological method. Biological method is otherwise called as green synthesis. They are using bacteria, fungi, yeast, plant extract. Here are some examples of biological nanomaterial synthesis. Sponge spike diatoms, reef, rice leaf blade. Biological synthesis. Rapid synthesis, controlled toxicity, controlling on site characteristics, and eco-friendly approach. Crucial point before going to biological methods are what biological system can be used to bacteria, plant and fungi synthesis nanostructure. Can we do it extracellularly? What is the biochemical processes leading to nanoparticle formation? Is site control possible? Better monodispersity. How about nanoparticles? Shape modulation. Can we use extract of plant in nanobiosynthesis? Let's see what's the answer. Biological system for nanoparticles. Microorganisms such as bacteria, fungi, yeast, they may be either extracellular or intracellular, representing effective biofactor. Intracellular means inside the cell in cytoplasm or cytosol, and extracellular means outside of the cell, on the surface or between the cells inside a colony. Why do organisms synthesize nanoparticles? Major reasons involved in such synthesis is detoxification, which can be carried out by the following processes. Number one, modification in the cellular transport mechanism to restrict entry of the toxic ion into the cell. Sequestration of the toxic species within the cell or outside the cell. Activation of energy dependent efflux pathways to eliminate the toxic species. Enzyme catalyzed oxidation or reduction of toxic species to a less toxic form. These are some examples of bacterial biosynthesis. Bacillus cereus, that is silver of ranges 5, Bacillus thuringiensis, silver 10 to 20 ranges. Fungal and yeast system. Gold-silver alloys synthesis. Fungus Fusarium oxysporum. It has been found that initially gold is reduced, followed by silver, leading to nanoparticles of gold-silver alloy. Depending on the biomass of fungus used, one can changes the composition of the alloy nanoparticles formed from pure gold to pure silver with increase in the biomass concentration. Here are some examples of fungi and yeast biosynthesis of nanoparticles. Plants and plant extract as a tool for nanoparticles. They provide single-step biosynthesis process. The protocols involving free from toxins and natural capping agents. They can generate bimetallic silver and gold cell nanoparticles. They are they have excellent stability and size control. They are cost-effective large-scale production of metallic, semiconductor and metal oxide nanoparticles. Here are some examples of plant extract nanoparticle synthesis. Green synthesis. Green synthesis having two important steps such as water is used as a solvent system and another one natural source of extract is used as a main component. Photosynthesis is implemented with plants as predominant source. It is a large-scale production of nanoparticles. They have two types such as extracellular and inexpensive reduction. Both microorganisms and plants have a long demonstrated the ability to absorb and accumulate inorganic metallic ions from their surrounding environment. Advantages of nanoparticles. Clean and eco-friendly approach as toxic chemicals are not used. Capping and reducing agent, large-scale production of nanoparticles, and also the experimental conditions like high energy and high pressure are not required. It leads to energy saving process. Application of nanotechnology. Nanotechnology plays an important role in medicines, pharmaceuticals, agricultures. It is excellent antibacterial, antifungal effect and anti-parasitic activities. It is very useful for environmental remediation and it is also the treatment of surface water, groundwater, fresh water contaminated by toxic metal ions, organic and inorganic solids, and microorganisms. It is very good pesticide control and management of plant disease. It is effective fertilizer and using for imaging, biosensor, gene therapy, and drug delivery. Finally, it can be concluded that production of nanoparticles using extract from natural substances is emerging as an important area in nanotechnology. Nanoparticles used for production of natural resources is sustainable, eco-friendly, inexpensive and free of chemical contaminants for biological and medical applications, where purity of nanoparticles is of major concern. The waste products of plant extract are non-toxic and easier to dispose of. Nanoparticles are furthermore synthesized by green route are more stable and effective in comparison with those production by physiochemical methods. Thank you.